This video is going to talk about the phenomenon called osmosis. This is a pressure that builds up as a function of concentration difference between two solutions separated by a membrane that lets only one of the parts of the solution through. Now, the most common example of the solvent being water, and the membrane lets water through but not the solute, such as salt or sugar or something like that. Our cells are made up of membranes that are very selective about what goes in and out. And so here's a picture of how you could measure osmotic pressure. So the little pink dots denote solute. And so we have a very concentrated solution in the middle and a not so concentrated solution on the outside. And so these two solutions are not equal in energy. This actually has a higher energy. And so what nature wants to do, of course, is to have both solutions be the same concentration. If you didn't have this membrane, then all the solute would spread out and then be spread throughout both liquids. But since the membrane will not let these solute molecules out to dilute the surrounding solution, what happens is the solvent goes into the more concentrated area to try to dilute it. And because it does that, then of course there has to be an expansion of the volume, and it's usually up a capillary tube. And the capillary tube, as it grows in height, increases the pressure exerted by the height of that column, just like we discussed in atmospheric pressure. And of course they'll reach an equilibrium, and that equilibrium pressure is the osmotic pressure difference between the inside and the outside solution. That's equal to the pressure difference of the height of the column of liquid. Okay, so our red blood cells have things dissolved called osmoles. Osmole is just a generic term for anything that creates an osmotic pressure when it's dissolved. And the most common thing to think about is sodium chloride, but there are other things that also produce osmotic pressure, such as glucose in the blood. So so if you put water into the bloodstream, you have more or less, this would be like the red blood cell, and this would be like the surrounding. Then water is going to go into the red blood cells and make them blow up. That's called the solution's too concentrated relative to what's outside. And so this solution out here doesn't have enough solute. It has too much water, and that's called hypotonic. Okay, so in a hypotonic solution, the water outside the red blood cell, it's going to go into the blood cell and blow it up. The reverse situation happens if you were to drink seawater, for example. Seawater is toxic not because of the identity of the ions, but of their concentration. And so basically, if you drink seawater, the salt concentration inside the red blood cell is now lower than the surrounding bloodstream. And so actually the red blood cells lose water and they shrivel up. And so the salt concentration is too high outside the blood cell. That's called hypertonic. So it'll actually suck water out. And this is actually what happens with pickles and so on. So if you remember, cucumber is mostly water. You put it in the vinegar solution, and so water comes out of the cucumber, and actually some of the other stuff goes into the cucumber as well. Okay, so we're just going to work a few questions here. It turns out that blood and other cells have an osmolarity of 0.3. And we're going to abbreviate that osmolar. Okay, so basically that is how many moles of particles are dissolved per liter of solution. Okay, so one osmole is, 1 osmolar equals 1 mole of particles in 1 liter of solution. Okay, and so most tissues in our in our body are 0.3 osmolar. So what that means is if you introduce a solution that is higher, that's hypertonic, that's bad. If you introduce one that's lower, that's also bad. It's hypotonic. So hypotonic is below, hyper means above. So we're going to ask ourselves, and it turns out that in salt, it's 0.9% grams per 100 mils. And so this is kind of a mixed concentration unit of grams per volume. And so this is the amount of sodium chloride that has the same osmolarity. Okay, so this is 0.9%. Mass to volume. All right. So let's look at a solution that is 2%. Okay. Well, 2% mass by volume of sodium chloride is hypertonic. Okay. It's too high. It's higher than the 0.9%. Okay, so 0.5% is hypotonic because it's below the 0.30 osmolar that you get from 0.9%. Now, what about glucose? Let's say we have a 1% glucose solution. Now, in order to do this, we actually have to calculate the osmolarity rather than just rely on what we know from sodium chloride. So with 1% glucose, you have 1 gram of C6H12O6 in 100 mils of solution. Okay, I'm going to go ahead and convert the mils to liters. All right, and so now we need moles of solute particles versus liters of solution. So where do you got the liters of solution? So we need to change the grams to moles, and that's using the molar mass. All right, and so this will tell us what the osmolarity is of 1% glucose. So that's in a decimal number, it's 0.056. Or 0. Okay, now wait a minute. Yeah, it is 0.05. Sorry. Okay. So we calculate this and we get 0.056 osmolar. Okay, we have mole of particles, which is for a non-electrolyte, just one for every molecule. And so we get 0.056, which is less than, so this is hypotonic. Okay, so we could also look at 5% glucose. With 5% glucose, this number will be 5 times higher, and it'll be very close to 0.3 osmolar solution. And so what that means is the isotonic solution for salt will be 0.9%. For glucose, it'll be 5% mass to volume.